What's happening Wednesday? Midterm. Good. We're all going to be on time so that everyone gets to finish appropriately and actually get to take the exam. So, but before that, of course, we have to have our clicker questions. No, the exam's not written yet. It will be by Wednesday morning, hopefully. We shall see.
Maybe it's not written by Wednesday.
That's okay. You'll manage if it's not done by then. Okay, let's see if we can actually get this to advance here. Here we go. So our first clicker question for today is: the organization of promoters in the SV40 genome is most similar to the arrangement of promoters in HIV-1, HPV, bacteriophage lambda, Ebolavirus, and poliovirus. Oh, sorry.
Okay. That's what lambda is. I remember we talked about lambda. Because that's what lambda has left and right. Yeah. So it's not
Yes, like lambda.
Okay.
I knew the answer.
10.
Oh, yeah.
0. So this is kind of question which is quite likely to be on the exam. People always ask, is our exams comprehensive? No, not per se. But I do assume that you remember at least some of the material from the previous exams. And so that would be like one particular one of these, like bacteriophage lambda, yes, has divergent promoters, just like SV40 does. HIV-1 basically has 2 promoters, but one of them's used. Which one is that? Where are the promoters in these HIVs? HIV-1, I should say, but it's true for all the HIVs. LTR is the promoter, and one of those is being used. They're both in the same orientation. HPV, they're also both in the same orientation, but they're right next to each other, which is kind of bizarre, but that's the way they are. Ebolavirus, negative strands, start-stop, transcription. Poliovirus is way too long ago that you were studying. Well, it's even further long ago that you were talking about bacteriophage lambda. So, yeah, promoters, again, for these RNA viruses doesn't really make much sense because there's not really a promoter in the classic sense in terms of the way these things work. So, yes, it is C because, hang on, let's not do that. Let's actually show the results for the last one. Okay. I'll get the correct answer for that anyway. So, we don't need to redo this one. We'll go to the next one. Obviously, my coffee wasn't working properly this morning. So our second clicker question, large and middle and small, should be a common there. T antigens in SV40 all are made from the same primary transcript, have the same N termini, have the same C termini, A and B or A and C. Ten. 3, 2, 1. Done. So, most of us agree, not quite everybody. Yes, they're all made from the same transcript because that's the, which transcript? P early, exactly, coming from P early, one of those divergent N termini, C termini. And why is that? Okay, they all start at the same place, but how are they different from each other? Why are they large, middle, and late? As small. Alternative splicing, but splicing happens internal to the open reading frame. And that means they have different C termini. So let's actually see if I can do this one properly this time as opposed to last time. I can do this manually afterwards, so don't worry. All your answers for last time will still count. So, yes, reading frames. It's all about the reading frames, correct. So, last clicker question before the midterm. Tumor formation by HPV is due to E1 and E2, E2 and E3, E4 and E5, E5 and E6, E6 and E7. Yes. True. F, the button down here. 10, 5. Yes, you can obviously go back and read your notes from last time. It is E, that's correct. It is E6 and E7. We'll talk about that towards the end of today's review once we get there. Yeah, as usual, this is review. This is stuff that either I thought I did a crummy job explaining last time or things that I think were particularly important. Again, no guarantee if any of these things end up on the exam. I have always thought it might be interesting to go back and look, compare the exam to what I did in the review session and see how well they correlate with each other. So way back when, yes, poliovirus. We did talk about poliovirus after the last midterm. These are now our RNA viruses, and most of the viruses we've talked about in this section of the course are RNA viruses. This one, of course, is very different because, to start with, at the 5' end, it has a protein, VPG, which is really important for replication that we'll take a look at again a little bit later on. Polyprotein, of course, so one RNA, and that one RNA gets made into one ginormous protein. That then gets chopped up into smaller pieces by the viral proteases. The other aspect, and why I've got the red circle on here, is that it's a poly-A tail at the end of the genome, but that poly-A tail is encoded in the genome. And so what's packaged, what comes in as a poly-A tail at the end of it, which is really quite different than most of the other ones that we've talked about since we talked about this, way back when. So probably the other really critical aspect, and again, big difference as far as these picoRNA viruses are concerned, are these irises, the internal ribosome entry site, present at the 5' the last slide, it was NTR, non-translated region. Most people call them UTR, so untranslated regions. So at the 5' end of the genome, you have a secondary structure in the RNA which can allow the ribosome to bind in the absence of the cap binding complex. And that's, of course, key to replication of these viruses because they can degrade some of those cap binding proteins due to the viral proteases and still get translation of your virus protein. And so that's not just in the case of these picoRNA viruses, but best known for these picoRNA viruses. So you get ribosome binding in a cap-independent fashion. Of course, you don't even have a cap in these guys because VPG is bound to the end. Why is VPG bound to the end? It has to do with how these guys replicate. VPG, again, is a short protein. It's one of those peptides that's part of the polyprotein, also known as 3B, which you can see over here. The 3AB starts there. You have first that protein which is made, then you have to add a couple of U residues to it. Why U? Because that's, of course, what pairs the poly A tail at the 3' end of the genome. Actually gets made at that Cre sequence right in the middle there. There are other secondary structures that are forming at the 5' end of the poliovirus genome, above and beyond that iris sequence we just talked about. Those bind to cellular proteins, but also the viral polymerase proteins. So the first couple of U's that have to get added, those are added by the 3D polymerase over here at the 5' end. So the step right before this one, up at the top here, is how the 3D polymerase will add a couple of U residues before it's going to bind to the 3' end and then make the rest of the genome here. So secondary structures and the 3D polymerase making a couple of U's before it goes to the opposite end of the genome. Once you have anti-genome RNA, then you really basically have no problem. You're not trying to deal with the ribosome going through in the opposite direction. So that's, you make much, much more of your positive strand than you do of your negative strand. Questions on these picoRNA viruses? Way back when? Yeah?
The membrane vesicles, so this all happens.
Oh, so the question is, where are these happening? Where's this replication happening? So membrane vesicles is really common. Lots of these viruses are replicated in membrane vesicles, probably to help to concentrate the proteins and the nucleic acid. It's all going on in the cytoplasm. And one of the proteins, I can't remember what it is, so I don't expect you guys to either, in the poliovirus polyprotein blocks vesicle transport. So they can't get vesicles out to the membrane, so you accumulate vesicles in the cytoplasm, so you have more places that you can replicate. So, but they're all cytoplasmic vesicles. So what happens if you put an envelope around these naked particles? You get Flaviviruses. We mostly care about Flaviviral diseases. Who heard about Hep C this morning on NPR? Am I the only person here who listens to NPR? That's sad. Talking about the really high price of those antiviral new medications. And so it was an interesting story. Apparently, a lot of private insurances and Medicare are not wild about paying for it. So there's some clear issues there. But basically, these are really picoRNA viruses that have an envelope around the outside. Yellow fever was a really nasty disease. Dengue is now clearly a really nasty disease. And Hep C can be well treated, but it happens really late in life. And whether treatment's such a good idea at that kind of expense is really a ethical and moral question rather than something we'll be discussing in this class. All these, except for Hep C, are arboviruses. They're being moved around by mostly mosquitoes, but biting insects in general, that are taking blood meals. What's on the outside of these particles is a really different, at least different looking fusion protein. Sits down flat on the outside of these particles. They really don't look like your coronavirus, anything else sticking out on the outside. Very flat structures, but still, when you undergo the pH change that you have in endocytosis, then you have rearrangement of these into your typical trimeric form that sticks out, which has these fusion peptides down here, which then stick into the membrane, and you see nice membrane fusion that takes place. Once you have membrane fusion, of course, you release your genome. That genome, again, looks a lot like these picoRNA viruses with a couple of differences. One is down here at our 5' end. We have a cap, but it's a positive strand RNA virus. It's replicating the cytoplasm. That cap is made by viral proteins. Doesn't have a poly A tail down at the end here. This almost always has a secondary structure, which is basically serving the same role. So it's protecting the 3' end from getting degraded. Also, this sequence is complementary to this sequence over here. So you have a loop that forms in your RNA, which again is very often what you see with cellular messenger RNAs, poly-binding proteins associating with the cap. So that's what this structure looks like. Again, polyprotein, viral proteases which are involved, and in fact, those new drugs are targeting the viral proteases and the viral replicase. The big difference here, of course, is, again, that we have envelope, and that envelope is being put together in a membrane, and to use that appropriate assembly process, you also need cellular proteases. So picoRNA viruses, pretty much, again, a few exceptions, always exceptions here, but for the most part, it's all going to be viral proteases. For the Flaviviruses, now you need some host proteases, most of them having to do with the formation of these membrane proteins. So signal peptidase, the furin peptidase, both of those are required to get your matrix protein and envelope protein appropriately processed so that these can be sitting in the membrane and giving you excuse me, the appropriate assembly process. So viral proteases down here, NS3, NS2B, signal protease or signal peptidase for membranes, and then the furin, which again we've seen multiple times now, is a classic peptidase. It's involved usually in processing proteins, either going to be secreted or going to end up at the cell envelope. Also, many of these viruses are using that particular protease to get final virion formation. So any questions about the Flaviviruses? Yeah?
So the Flavivirus is in an envelope?
Yes.
In a membrane. How does it stay in an envelope once it leaves the cell? My question is, how does it get transported without losing that?
Right. So the question is, sorry, I'm just repeating it so it's on tape here, but the question is basically, how do you have something which is enveloped, and that envelopment is taking place here in the endoplasmic reticulum? How do you keep that envelope once it gets to the outside of the cell? And the answer there is that these end up forming multiple layered membranes, and so there's going to be a membrane on the outside of these envelopes. And so that external membrane will then fuse at the plasma membrane, releasing the membranous structure which is on the inside. And there's some pretty nice figures in the textbook that show that. Other questions on Flaviviruses? Okay, so getting bigger, we go to the coronaviruses. And this is also a nice way to introduce and talk a little bit about some of the questions that have been on the discussion board on D2L. A couple of you asked me about porcine transmissible gastroenteritis virus. So this is the one that's causing pork prices to go way up, lots of piglets to die. It's a class 1 coronavirus. And one of the big questions was, well, why is it not causing nasty disease like our friends down here? It's probably pretty similar to a lot of these cold viruses. So yes, sure, it can cause some disease, but generally not a major problem. On the other hand, of course, these class 2, and particularly our friend SARS down here, do cause nasty disease, and so much that people get really excited and scared about it. Absolute numbers of SARS cases, less than 1,000. So yes, something to be concerned about, but probably not this concerned about. I think much more importantly, in terms of SARS and SARS epidemiology, what has hopefully gotten somewhat through to the public is that these are viruses that are circulating in other organisms and probably not causing much in the way of disease. So we like to think of these as the reservoir species, where the virus is replicating apparently perfectly normally, being maintained in not so much a population, but really the ecosystem. And then occasionally that will then spread to some other organisms, and usually because you've got them in too close contact or you're ruining their environment. There are lots of other reasons. You'll hear more about that next week as well after the midterm. So basic idea here is we've got huge numbers, all kinds of different bat coronaviruses. Again, sequence differences relative to each other, but the ones that are causing disease in humans are also probably pretty clearly bat viruses as well. What's in these viruses, virions, is this really huge single-stranded RNA, positive strand RNA, also made as mostly polyprotein, but all of these structural genes down here at the 3' end, which has a 5' cap and a poly A tail, again, made in a very similar way to what happens with the Flaviviruses. These are all viral proteins that are involved in this. Here, the formation of these structural genes is due to the nested subgenomic RNAs, which are being made, and we'll talk more about that a little bit later on. As normal for these large single-stranded positive strand RNA viruses, there are viral proteases which chop up polyproteins in order to give you reactivity. What's different in terms of the coronaviruses is all of them have these separated open reading frames, and sometimes you get translation of these extra enzymatic proteins here, sometimes you don't. Usually it's more of just this first part, and that's due to a secondary structure in your genome that causes a change in terms of translation. Where else have we seen this? Changes in translation, open reading frames due to secondary structures. A couple of different places. So RNA editing that you see in the negative strand RNA viruses, but also what happens in the polyprotein in retroviruses. Same thing, also get frame shifting that takes place here. So it's a completely different viruses, but a very similar kind of mechanism, which is there. Of course, the other mechanism here are these nested subgenomic RNAs, which are made by the RNA-dependent RNA polymerase. Every time it gets to one of these basically pause positions in the genome, it can decide whether to jump to the opposite end of the genome or keep going. That gives you many more of these subgenomic RNAs down at the 3' end of the genome, which is where your structural proteins are. And of course, we need way more of those structural proteins in order to make the whole virion structure that you end up with at the end. So here, many more of these subgenomic RNAs than you have of the full-length genome RNAs. Questions on coronaviruses before we flip strands? Go to the negative strands. Okay, so now I wanted to talk about the Paramyxo and rhabdoviruses, which are basically the same, only with a slightly different structure. Here's our cartoon of one of these Paramyxoviruses. Again, negative strand RNA viruses. You've got to bring the kitchen sink with you in order to make more of your virions. So packaged inside the virion is not only their nucleic acid, but also your polyprotein, everything that you need to at least make that positive strand RNA in order to be able to make proteins to make more of your virions. So the inside is really different. The outside is something a lot similar to what we've seen before. We've got hemagglutinin neuraminidase. In this case, it's one protein binding to sialic acid, chopping off that sialic acid. Then there's a separate fusion protein in the case of these Paramyxoviruses. In VSV, the rhabdovirus, both of them are together as the same protein. But this should also seem really similar relative to what other kinds of myxoviruses? Orthomyxoviruses, which are influenza. So influenza HA basically has the hemagglutinin activity and fusion protein in one, and then a separate neuraminidase. But again, the activities here are basically identical. They've all got the same problem. They've got to get into these, and these are all respiratory viruses getting into the epithelia, although it's not quite true. Rabies is a little bit less so of one. What comes in, again, is a negative strand now RNA virus. These are the mononegavirales, so one negative strand is the genome. Now, of course, we flip things around, 3' over on the left, 5' over on the right. What you have are these interesting intergenic sequences between all of your structural proteins, which you have down here at the 3' end, and then the non-structural protein, the replicase protein, down here at the 5' end. What happens here is you start making your copy of your genome, all negative now, positive strand for each of these individual little segments. After you get to the end of the first one, you stutter for a little while, add a poly A tail. Sometimes you fall off. Some percent of the time you stay on. Get to the next position, stutter a little bit, sometimes stay on, sometimes fall off, and eventually get down to the end of the genome where you make more of the replicase protein. This is great for making messenger RNAs. It's really crummy for making genomes. So at some point, you've got to switch from doing this stop-start stuttering thing to making whole copies of the genome. That's all dependent on the concentration of the N protein. So how much of the N protein you have binds to your template. Binding to the template will then lead to replication of anti-genome and then full genome in the absence of stuttering and stopping and restarting all the time. The only other thing we wanted to talk about here, particularly for these Paramyxoviruses, is the stuttering that happens inside protein coding genes, also known as RNA editing. So here, the addition of extra nucleotides in polynucleotide stretches is going to change the reading frame of the encoded protein, which is going to change the C-terminus of that particular protein. Also present here is the utilization of different start codons, which will give you multiple different proteins here in the same piece of the genome. And as we talked about when I had that wrong answer to one of the clicker questions, these are also involved in pathogenicity, these extra proteins here. Whereas the P protein is part of your polymerase. So it's a phosphoprotein, which is where the P comes from, but it's part of the replicase complex, which is involved in making more of these. So there are questions on these extra questions, Paramyxo, rhabdoviruses, very similar again to each other in terms of these replication, the molecular biology thereof. Okay, we stretch one of those rhabdoviruses out. We get a filovirus, big, bad, nasty disease viruses, but basically molecularly they're extremely similar with, again, a couple of interesting differences. One of these is the GP glycoprotein, which we'll talk about in just a second when we talk about the editing. The other one is the VP30 transcriptional regulator that's involved in regulating transcription due to secondary structures that form in these genomes. The arrangement of the genome, again, mononegavirales, so single molecule negative strand with your structural proteins at the 3' end, non-structural proteins at the 5' end, start, stop. Very normal, again, down here at the bottom, we've got our Paramyxoviruses. Difference here is that sometimes your start and stop overlap with each other. Okay, why that is not entirely clear, but they do, and it's true in all of these filoviruses. The other thing, and probably much more important, and particularly in terms of disease, is the stuttering and RNA editing that happens in the glycoprotein, the GP. Again, that GP basically serves the same role as the hemagglutinin neuraminidase and fusion protein. So all of those things combined into one protein. In all of these filoviruses, so not just Ebola, but also Marburg, you have stuttering that happens, which leads to most of your glycoprotein being secreted, so not bound to the external membrane of your virion, but small amount actually then present in the virion. So that's the big difference that we have here. Again, it's still a change that happens in the C-terminus of the protein. Yeah?
What was the function of the secreted glycoprotein?
So the question is, what's the function of the secreted glycoprotein? Yeah, so again, what the function of it is still very much open to debate. And whether it's critical for pathogenicity, it's hiding the virus from the immune system, is really not entirely clear. And that's partly why people have made these proteins like this one down here at the bottom. We've changed the genome such that you don't have secreted glycoprotein. They're trying to see what that does in terms of viral disease. And there have been a couple of papers that have been written on this very recently. TWIV talked about it, I think, a week and a half ago. But yeah, at this point, it's still pretty unclear exactly what that secreted glycoprotein is about. But clearly, you do have a lot more of that than you do of the one that's associated with the genome. Or I'd say associated with the virion, I should say. Yes, they do cause nasty disease. 60-odd% fatality. Unfortunately, lots of these people are the first responders, the healthcare workers, but the absolute numbers are really still very small for these diseases, probably very self-limiting, mostly because, to quote Ian Lipkin, talked about this for MERS, but the same thing is true for these viruses. They're wimpy viruses. They have a lot of trouble getting from one human in particular to another human. So you need very large amounts of virus and usually direct bodily fluid contact. Questions on the mononegavirales? Again, most of which are pretty similar to each other. Now let's look at our friend influenza. Taking that humongous genome, splitting it into multiple pieces, makes a lot of sense in terms of regulation. So you can have each of those individual pieces be making a different message, giving you different messenger RNA, but gives you the problem of having to put all of those individual segments into a particle in order for that particle, the virion, to be infectious. So that's, again, still a very open question how that works. The other thing, which is what I think I've neglected to mention, at least not sufficiently, I think, when I talked about this in lecture, is that because of these individual mini chromosomes, as I like to think about them, segments, as the virologists normally call them, since you've got all these different segments, if you have co-infection with two different influenza viruses at the same time, you can mix and match each of these guys. And that's what leads usually to these pandemic strains, things where you have a big jump from, say, an H5N1. One of these would be encoding your hemagglutinin. The other one would be encoding a neuraminidase. if you now swap these out with different viral genes, then the immune system, which is mostly recognizing these proteins here on the outside, is not going to be recognizing those particles anymore. And so that's where the real problems and worries are in terms of emerging new influenzas, and then the swine flu, which only had 2 segments from the swine and 1 from avian, and the rest of the 5 were human. That was one of these cases where you had this reassortment, as people call it. So reassortment of those individual viral segments or individual viral genome pieces. Messenger RNA production by these real or orthomyxoviruses. These guys replicate in the nucleus. The reason they're replicating in the nucleus is because that's where the caps are. Because they're using capped messenger RNAs to be producing each of their individual proteins. And those caps are snatched, according to the official literature, stolen by the viral polymerase proteins. And so those will come in and cut off the caps from cellular messenger RNAs, which are being kept in the nucleus because there are some viral non-structural proteins that block normal tailing. So you end up with accumulation of these extra messenger RNAs in the nucleus so that, no, not so that, evolutionary speaking, is taking advantage of the fact that this is true. The viral polymerases, PB1 and PB2, can bind to and cleave these cap structures and then start using those as primers, basically, for doing the rest of your replication. And so this also gives you a pretty good idea what kinds of polymerases, what the structures of these polymerases are. They're basically DNA-like polymerases. They need primers. And so that primer is then bound to that. This is part of the cap, and then you replicate down to the other end of the genome. This, of course, is what happens in replicating messenger RNAs. You get stuttering, just like what happens in terms of the other negative strand RNA viruses. The way you switch from stuttering to making complete genomes is presence of your nucleocapsid protein. As soon as you have enough of that nucleocapsid protein, then you flip around and stop stealing caps, start at the complete opposite end of the genome, and then replicate you through the rest of the way. So those are the big differences. Again, it's mostly having to do with these multiple different pieces and the cap snatching process. Briefly revisit vaccines. Somebody asked about, okay, if the same vaccine is recommended for next year as it was for this year because the match was really, really good this year, do you still need to get vaccinated? I have a couple of emails out asking about this, but at least the CDC says that your immune response decays over that 12-month period, so it's a good idea to do so, but I will definitely let you know what's going on as far as that's concerned. A couple of other quick things on here as far as vaccines are concerned. One is that it's a mixture of different viruses. Again, H1N3, N1, N2, at least in this year's, and again, most of the viruses circulating this year. This H1N1 is extremely similar to the swine flu that we had in 2009. And so that's the main circulating form that's going around right now. The influenza B, trivalent vaccines have 1 B, tetravalent vaccines have 2 Bs in them, all of which, again, were really quite good matches as far as the particular vaccine was concerned. So questions on influenza, etc. Okay, let's talk about retroviruses. Retroviruses are sort of in between the RNA viruses and DNA viruses, partly why I put them in this particular part of the syllabus. You'll notice they're jumping around in terms of the chapters in the textbook. But here is the genome of one of these, or so the genome that's packaged inside a virion of a classic retrovirus. We'll talk about some of the other ones later. Cap and tail, all cellular, because this is the cellular polymerase which is making all of these genomes. A repeated sequence, and then unique sequences at the 5' end and the 3' end. Then a primer binding site at the 5' end and a PPT down at the 3' end, which of course we all remember is also going to serve as a primer. For all retroviruses, you have a major 5' splice site and 3' splice site. That splicing generates the envelope proteins, which are present on the outside of the virion. And these, again, are the receptor binding proteins, fusion proteins, which are involved in getting that attached. They're also made as polyproteins, like we've seen for all of these other positive strand RNA viruses. Also overlapping open reading frames, like you see in Virus 174, but more importantly in terms of the positive strand RNA viruses that we just talked about. Also frame shifting, multiple frames in polyproteins. Mutter, mutter, mutter. Starts with S, 4 letters. SARS, which is a coronavirus. So coronavirus is extremely similar in terms of this frame shifting that takes place here. So polyprotein is made, viral protease. One of the big differences that you have with these retroviruses is this protease is active very, very late in the whole virus assembly process. Basically, it's only being used very much at the end when you're finally putting all of the virus particles together, and in some cases even after that virion has budded from outside the cell. So these are our structures: matrix, nucleocapsid, and capsid, protease, reverse transcriptase, and integrase in terms of the GAG, POL, and ENV proteins. Again, unfortunately, the terminology is not great. Group-specific antigens, polymerase, which is all the enzymes, and the envelope proteins. Most important, as far as these retroviruses are concerned, from a molecular point of view, is, of course, how they're replicating. When the genome comes inside the cell, up here at 1, it also has a cellular tRNA bound to the primer binding site. The reverse transcriptase, which is present in the virion and is active in the virion, will use that primer and extend from that primer binding site out to the 5' end. That's our minus strand strong stop that we mentioned in the clicker questions last time. The reverse transcriptase, in addition to its polymerase activity, also has an RNase H activity. RNase H cleaves RNA and RNA-DNA hybrids. That then chews off the end here after step 2. And so now you have a complement to your repeated sequence and the unique sequence at the 5' end. The complement to this repeated sequence that you have at the still actually would be the 5' end of your genome, that's the 3' end of the antigenome, is of course complementary to the repeat sequence that you have at the not quite exactly 3' end, because there's still a poly A tail hanging off the end here as well. That binds here after your first strand transfer, and then you get synthesis all the way through to the opposite end of the genome, stopping at your primer binding site. RNase H does its thing, chews up the genome, except for the PPT. And the reasons that the PPT is not being degraded probably has to do with some secondary structures. It's a little bit more resistant than the rest of the genome. That then serves as a primer for your positive strand, which, again, bound by the reverse transcriptase, will use now the DNA template. So it's not a RNA-dependent DNA polymerase now, it's a DNA-dependent DNA polymerase. Replicates out to the position in the tRNA that is no longer recognized by the reverse transcriptase. Turns out it's a pseudouridine, which is always present there. And what do you match with a pseudouridine? You actually can't add a nice nucleotide to a pseudouridine. So it leaves opposite. So that's where that stops. This basically gives the RNase H a little more time to chew up any kind of RNA that has secondary structure. Clearly, tRNA has a whole bunch of secondary structure, and our PPT has secondary structure. Now we've got a complement to, well, in fact, the original primer binding sites, the complement to the tRNA, which can now, of course, bind at the opposite end of the genome or the other genome. Again, remember, we've got 2 genomes in all of these retroviruses. That then is your second strand transfer, provides templates and 3' OHs, so you can extend and generate 2 LTRs, one at each end of your genome, where you have repeated sequences, which you'd had in your normal virus, and then you've added the 3' unique sequences to your 5' end and your 5' unique sequences to the 3' end of your genome. So any questions on this whole molecular, I don't know, gymnastics, I like to call it, of getting these genomes to replicate? Pretty amazing, at least to me, how these things have managed to do this process. Okay, so other aspect of these retrovirus genomes, as I mentioned before, we've got in all retroviruses splicing between a major 5' splice site and a 3' splice site, and that's all for generating these envelope messenger RNAs. These are going to get translated in the endoplasmic reticulum. Those membrane proteins are going to get transported to the plasma membrane. That's where you're going to end up with putting your whole assembly together. You also have full-length RNA, which is made. You have to have enough of that full-length RNA to get it packaged and also to make the GAG and Pol proteins. This is pretty inefficient for most of the retroviruses because you end up with a mixture of your spliced RNA and your unspliced RNA. HIV has figured out a way, i.e., again, through evolutionary processes, and that is these extra proteins which are involved in HIV. All of these are generated through yet another round of splicing that takes place. So main ones that we talked about are the TAT and Rev proteins, which are down here at the bottom. These are generated by 2 splicing events. The main splice, which happens at your standard 5' splice site, and then differential splicing here, but the main point is TAT and Rev, which are then being spliced separately. TAT is your transcriptional regulator, which leads to transcription, basically under the appropriate conditions in the cell cycle, when you can make lots of, and TAT also doesn't just regulate full-length RNA, it also regulates some of these other RNAs, so it's about 100 nucleotides that the polymerase will get to after starting in the LTR over here. Presence of TAT, you're going to make a lot more of the RNAs. In the absence of TAT, you make a lot less. The Rev protein is the one which guarantees that you get full-length RNA being transported out into the cytoplasm, where it then gets brought to the membrane and enclosed in virions, which then get released and go off and infect other cells. Questions on retroviruses, HIV, et cetera? Yeah? What's doing the splicing? They're all cellular proteins, so all cellular splicing proteins. And so they are very normal, as it were, binding sites for the U1, U3, U4, U5, U6, standard RNAs in terms of the spliceosome. In fact, a lot of what we know about splicing has been looking at splicing in some of these virus genomes. So splice enhancers, for instance, there are a number of splice enhancers that are present in here as well. So it's not viral proteins. The viral proteins are not involved in splicing. There are some viruses that do have that, but we're not talking about those in this class. Okay, so the last set of viruses that we talked about since the last midterm are these small double-stranded DNA viruses, which are marvelous tools for understanding transcription, translation, etc. in normal again, quote unquote, DNA because it's all packaged in these histone structures. What do you see in that genome? We already talked about this in our clicker questions. Divergent promoters, early promoters, late promoters in the polyomaviruses. The early promoter leads to production of one transcript, which is then differentially spliced, giving you the various T antigens, small, medium, and large, and in some cases even the tiny one, but that's not as critical. The late genes are made from a number of different promoters. The splicing that happens there happens before you get into the coding region, and so these guys don't have the same M termini as each other. The last thing about these genomes is that their transcripts end up overlapping at the end of the genome, being a relatively small circular genome. That generates double-stranded RNA, which leads to degradation of transcripts. Probably most important is the degradation of the large T transcript. But the large T antigen also blocks transcription from its own promoter. Yeah?
With respect to the overlapping terminators,
yeah.
When you say that that makes for double-stranded RNA, is that just a result of the complementary pair bonding between the two ends of the early RNA transcripts and the late freshly made RNA transcripts?
Yes, exactly. So the idea there is that if you're still making early transcripts, then it's actually probably too early to be making late transcripts. So the process of making early and late transcripts that overlap with each other, generating double-stranded RNA, means we're probably not at the right place in terms of making a lot more of these particular viruses. So, yeah.
Conceivably, is that meant for this to happen because the RNAi will actually, mechanisms will take care of it and degrade them?
Yeah, so that's exactly. So that's the idea here, is that the RNAi mechanisms will then block the transcription of either of these, because it's going to be both, if both of those two strands. But they shouldn't be being made at the same time. If large T, there's enough of large T around, it should be blocking any transcription from the early promoters. So you shouldn't have any of these, of the transcripts coming around this side. Okay, so that's it for the polyomaviruses. The papillomaviruses have a very different gene structure. They have overlapping early and late promoters. The early promoters lead to the production mostly of E6, E7, and to some extent E1 and E2. E6 and E7 are, of course, the ones that are critical for the tumorigenesis of these viruses. E1 and E2 are really the sort of large T antigen equivalent of these papillomaviruses. They bind to DNA, they're serving transcriptional regulation, they're serving as regulation of your replication as well. So these are those particular proteins. Again, E6 and E7 we'll talk about in just a second. Probably done them almost to death now. But E1 serves as a DNA helicase, again, extremely similar to the large T antigen, and E2 is a transcriptional regulator leading to activation of particularly the late transcripts. Also is involved in repressing the early transcripts. So again, very similar to what's happening with the large T antigen. And is also a transcriptional activator, and I guess it doesn't talk about the transcriptional activation here. But all of these things are what's happening with the E1 and E2 proteins. E6 and E7 bind to RB and P53. E7 basically pretends that we're going through the cell cycle and releases the E2 factors, so those things that are being stimulated by papillomavirus E2 in terms of transcriptional activation. So these are being stimulated. They'd normally be blocked by RB. The RB binds to E7, then you're going to get all of this transcription leading into the cell cycle, which is great for the papillomavirus, generally not too good for the cell. Well, what normally happens inside the cell when you have things that are going wrong, P53 gets induced, will either block the progression through the cell cycle by expression of P21, or if things are really bad, lead to the induction of apoptosis. So it's not in the virus's best interest to have this happen. So E6 binds to p53 and degrades it. So E7 leads to this unnatural progression in the cell cycle. p53 should be blocking that. E6 comes in and gets rid of p53 so that it's not an issue. Any more questions? If you have any more, please email me. I will be checking that. Yeah, one more question in the back?
Is the influenza?
Yeah.
There's a lot of different charts about the progression and how well one is to know those.
So the question is, what about all the extra charts in this year's influenza, seasonal influenza? How much of that do I want you to know? Not all of it. I can't expect that. But what I would look at is sort of when you have individual peaks, you know, the time of year that this is usually being at the largest amount. Potentially something, and I haven't written the exam, so I don't know yet, but like 2009, that there were a huge number of deaths in terms of young people, way more than we'd had in previous years. But in terms of the individual details, you know, that there were 123 deaths in some year or other that I can't remember what it is, that I would say is not that critical.